Okay, the next speaker is going to be Dr. Joy Cavagnaro, and she is president of Access Bio.
Thank you, Ellen. Ellen introduced me that I had decades of experience, so she neglected to say that I was a child prodigy, so I have to just clarify that. So I'm going to talk now, transition now into the preclinical. And this is just to start off by saying that we have had much success in really navigating a path to curing mice of a lot of really difficult diseases. But our challenge today is really to bridge that path that the valley of death to the clinical trials. And so there are some directions for novel therapies, but the ways are not well marked. And I usually use the analogy that my definition of critical path is the reviewer. Whether or not it's a grant reviewer or an FDA reviewer, that's really your critical path. But I think in this regard, I might also point out that that lone person here could be the patient, right? And that's really our goal, to get to clear that path, to expedite that path. It's important to understand why drugs fail as we go forward, to try to do a better job. And this was a recent report by McKinsey, which you'll see that, in fact, most of drugs fails are not related to safety, although I think many people think that. In fact, it looks like most of the drug failures are actually that they just don't work. And why is that? Well, it's because the animals are not predictive, right? And that may be the case, but I would submit that in late-stage trials, what it really states is that humans are not predictive. So what are some of the challenges for novel therapies? I mean, I think we have two dilemmas. We have a preclinical dilemma and a clinical dilemma. So we'll start with the preclinical. I think it's schizophrenic use of data. We believe our efficacy studies in animals, and we challenge the toxicity. Oh, the species might be too sensitive, right? I think we clearly have an inefficient use of our pharmacology models. Many a time we're looking at a single dose. We don't explore the dose response. And God forbid we ever go higher, at a very high dose to worry about seeing any toxicity in our pharmacology models. And so we rarely include any safety endpoints. So it's really a missed opportunity there. I can always tell the difference between a pharmacology data slide and a toxicology data slide, because for the pharmacology data slide, there is never any mention of dose. There's a response, and that's it. And again, as I said, we're concerned about seeing toxicity in the toxicity studies. And for most, in most product development, toxicity studies are actually done in normal animals. And so we're asking our animals not only to extrapolate cross-species, but cross-physiological states. And so we'll talk about that as we move forward in some of the therapies that we're talking about, really the importance of considering the use of animal models of disease. And then finally, I think we design our toxicity studies to support a discipline. You do CMC, you manufacturing, you do preclinical, you do clinical. And we're doing our animal studies to satisfy a discipline. And I'll go forward why I think how important that it's not to do it that way, but to do it an alternative way. So now our clinical dilemma. So for novel therapies, by definition, they're novel, and there's high risk because they're unique. Novel is scary to the regulatory agencies. It's important for funding. It's very scary to the regulatory agencies. And for novel therapies, when we go into patient populations, they're the most dire, the most difficult to see efficacy, the most easy to see, to not be able to discriminate in terms of safety. So then the challenge becomes, well, you're going into the patient population, and you don't see an activity. How do you fund? How do you go forward in terms of clinical development? Let me just point out another point in terms of novel therapies. and I think it's more related to gene therapies and cell therapies as we are speaking about, is that the irreversible urge to improve. Drug development is not a constantly iterative process. As Keith showed you, I mean, it's very, and once you're on that chevron, once you're on, as Ellen also presented, once you select that lead candidate, that is your lead candidate. You don't keep tweaking along the way. And I think that that's a challenge because there's this urge to tweak, and a higher urge to tweak in academia, I think, than in industry. Industry knows, you have to lock down your process, and then you evaluate. So I think that's a challenge with novel therapies. And then the population that we go into is the disease population, or patients and not normal volunteers. So again, we need to show an active dose. And I think the biggest challenge for any, let's say, products that are being developed, and I say in the area of gene therapy, but I would say it's even higher in cell therapy, is this absolute ludicrous expectation that we may cure in that first trial. That is huge. That is an incredible misconception, but an incredible challenge for us in terms of designing our first-in-human trials to actually inform that we're not curing anybody in that phase 1 trial. We are trying to understand something that's safe and something that's active. But the major challenges, I think, are reflected in a review by Brian Kwan et al. And referring to the scientists, whose hard-fought, extramurally funded survival comes mainly from mechanistic, discovery-based science. And there's few resources to indulge in the opined importance of independently replicating experiments. That's a major challenge. And that the vast majority of scientists really can't even think about the concept of a large animal with respect to funding. And then industry sponsors have this intellectual property issues that may clash with scientific mantra of peer-reviewed dissemination. That's a challenge. And full-time clinicians who want to try as quick as they can to get something into patients may be a little bit impatient about the need to conduct animal experiments. And then, of course, there are the regulatory hurdles. So it's interesting, the comment was academic, questions about academic. And actually, in this area, the difference between academic or investigator-sponsored and industry-sponsored. And I think this is an interesting graph that the FDA put together in October. This was based on October 2012, actually, of the differences between INDs for commercial versus research sponsored. This is huge. This is not a graph that you would see for drugs, right? Nor is it a graph that you would see in the Office of Vaccines or the Office of Blood. This is the Office of Cell, Tissue, and Gene Therapy, which regulates cell, tissue, and gene therapy. And interestingly, the big disconnect between commercial versus industry research-based, investigator-sponsored. This is really stunning, this difference. And we can talk about, some expectations later on in the Q&A, if there's differences in expectations. Now, Ellen neglected to mention that I also was a regulator. So I refer to myself as a recovering regulator now. And so I want to share with you some of my favorite sponsor quotes as a regulator. One is when we have the pre-IND meeting, sponsors saying, we're talking regulatory now, not science. To which were those of us who are FDA reviewers, okay, what is that? And then I guess my most favorite is, my study is 70% GLP. Would that be okay? Okay. That's my one GLP discussion at this meeting. The other one is, this whole concept of GLP, and we could go into the expectations in the spirit of GLPs. And, of course, this really came to light post Jesse Gelsinger and the issues with gene therapy. And it got to be a little bit ridiculous because we got into the spirit of GLPs. Now you have to do full GLPs where academic labs cannot do GLPs and don't ever say you can. And it's completely ridiculous because even people who do GLP studies all the time get 483s or inspection notices. So, and then the design issue came up. I can't afford to do GLP. I'm going to not use this control animal because I can't afford to do it because it's, you know, it costs twice as much to do something GLP. So I had this statement, if compliance is good and compliance makes sense, can you have good science without full compliance? And the answer is yes. So we can talk about what expectations are GLP, but that's my GLP part of this lecture. Now my most frequent question as a consultant is, what's the least amount I have to do to get into the clinic? To which I retort, why couldn't you say what's the right amount? I mean, I'm not going to say what's the least amount. So what does that mean? So this is what I call, what do I have to do? So this is, did you do? Okay. What did you do? Did you do a 2-week? Did you do it? So, I mean, for pharmaceuticals, this is clearly, there's just something the expectations. There are guidance there, and most of it, what you'll see are required. So did you do a 2-week? Did you do a 4-week? Did you do a gene tox testing? That's very clear, right? For biopharmaceuticals, then it gets a little bit more towards the novel therapy aspects of it, and that we have more of maybe generally, maybe case by case, not done, etc. So I'm going to speak to this whole essence of case by case and not a did you do approach, but more question-based approach to designing preclinical studies. Now, I'll flash back when I was a regulator, and I was at NIH campus, and we had a meeting with NIH investigators. Some of you might recognize some of these names. That's me over here. This is Building 31, and sat across the table and basically was bombarded with, or arrowed or whatever, speared. FDA should not require animal studies to support first-in-human studies of human cancer vaccines. That's basically the discussion, and it went downhill from there. When giving one of the first regulatory lectures to the gene therapy community back in the early 90s, where we met at Williamsburg, I had a slide. FDA no longer requires preclinical studies to support first-in-human trials. I had the only regulatory talk, so I had to somehow get their attention, to which I basically said that. Now, to tell you that after my lecture, about a dozen people came up to me and said, I wrote that down, tells you where people's heads were, that FDA would actually even say this. But anyway, so if it were true, then my question to you is, how best do we go from plate to people or dish to dose? So I would like to focus, any questions afterwards is, do you recognize yourself in any of these? I would like to have people focus on dose. That the reason that we're designing these animal studies, as imperfect as they are, is only to pick a dose. And going into patients, we want to pick not only a safe dose, but an active dose, because we don't have an option like a phase 1 clinical trial that one does in normal volunteers, where we can have 6 cohorts of 6 or 12 per cohort. We may have 2 precious cohorts of patients, of 3 or 4 patients, to test and try to get an active dose. So when thinking about animal studies, I don't want you to think about how many species or how many studies or what. All I wanted is, what do I need to pick a dose in humans? Because that's really all we use these animal studies for, is to pick a safe and active dose. So if you don't have, I did the analogy because it's just like being a parent, right? You tell your kids you can't have something, and now they want something. So if you don't have any animal studies, or they're not well designed, then how do you design the clinical trial? Because again, I use the term preclinical. I'm very precise in it. I think non-clinical is very negative, and it tells us what we're supposed to do. Pre the clinical. This is why I'm doing the studies, not to do animal studies, not to cure animals. It's really to try to inform the clinic. So if you don't really have the studies or not, then you don't know how high to go. So you have to start very low. You don't know how fast you can go, so you have to go very slow. You don't know what to monitor in the clinic, so you have to monitor everything. You don't know how long to monitor it, so you have to monitor for a long time. You don't know who best to include. You don't know who best to exclude. So is this really faster? I mean, to the endpoints, to approval, to the patients, is it faster? And is it less expensive? Is it less expensive to answer all these questions and monitor everything in the clinical trial than to try to do something preclinically? So a paper came out recently on, genomic responses in mouse models poorly mimic human inflammatory diseases. You're right, we cure a lot of animals. And our efficacy studies are now being challenged. Here's a concordance paper actually done with monoclonal antibodies, which actually showed that the pharmacology was more representative of the clinic than some of the toxicities in terms of more predictive. And again, in this same paper, Kwan did the survey, but also looked at translational research and what's needed to support to go forward in spinal cord. And interesting, surveying researchers in the field whether or not single species, multiple species were needed, which animal models were best, etc. And then last year, the NIH had a symposium, again, on improving animal models for regenerative medicine. And so we are trying our best to try to improve our animal models, again, so that we can pick a dose for the clinic. In terms of preclinical toxicity, there are a number of toxicities that we see in animal models, again, that are more sensitive. So if we see a toxicity, that doesn't necessarily mean we stop development or we can't move forward in development. But I say we modify clinical development or modify the clinical trial design. So if you're looking at a monoclonal antibody and you have cross-reactivity to endogenous molecules or tissues, we may modify. If you have a narrow therapeutic index, we may modify. If you have similar toxicity across species, so if you have occasion to do more than one species and you have similar toxicity, then you can't say that one species is more sensitive, then you have two species with that toxicity. If you have difficulty in monitoring any organ, so if you have toxicity in the choroid plexus, well, you can't monitor. It's different than if you have liver toxicity where you can monitor. If we see any delayed toxicity, infection, neoplasia, tumorigenicity, then there's a higher level of concern or modification for the clinical trial. Again, transmission of infection or disease. And it's often thought if we have enhanced toxicity, again, with duration or with the disease model, we might consider modification to the clinical program. Not necessarily stop, but modification. So then what happens when there's no relevant species? I mean, technically, we could say that for many of our cell-based human therapies where they're all xenotransplants. So we can use transgenic animals, right? So that's not a disease model, but where we try to humanize our animals. We might humanize them with a receptor, and then we can use the clinical candidate. So if you had a monoclonal, you could humanize it, and then you could use a clinical candidate. If we're developing a growth factor, we might have a knock-in model, and that way to look at worst-case scenario overexpression of a growth factor. Again, knockouts would be the opposite, worst case, but of maximum inhibition of that. Again, but there have limitations. And I think another key message in the lecture is just as humans have limitations as well as predicting in the late stage trials, we start off with every single animal model has a limitation. And we just have to do our best to understand those limitations and consider those limitations in our design and also in our interpretation. Because at the end of the day, then we're using these models based upon what we know to help us, inform us in terms of how best to extrapolate the data that we generate. And so, for example, if we look at differences in epitope density, if we're talking about monoclonal antibodies and localization, compartmentalization of cells, or turnover expression of the, receptor or signal transduction pathways, all of those what we know, similarities and differences within the animals, that helps us in terms of our extrapolation of the data. And then we have the use of animal models of disease, which is, again, probably most used in our cell-based therapies. This is the area where we take advantage of these disease models. Now, in terms of standard did you do toxicology or developing a small molecule or, for that matter, a large molecule, generally disease models are not required or at the same level. And I think some of the reasons why disease models are not used is because they're variable. They may only mimic one aspect of the disease, and animals might be too sick, and so you can't discriminate in terms of toxicity. Clearly, the historical data is much less in various animal models of disease than they are in normal animals. Certainly, the increased sensitivity may not be relevant, and so we're challenged with that. And it may otherwise, one, you might not be able to have, there might not be one available, or it might be very expensive or, resource intensive, or you might only be able to have a few animals. And so those are somewhat limitations in terms of the disease models. But then I think there are advantages. And again, you might have a better idea of exposure or activity in the subject you're treating that is disease. And you may have a better idea of the therapeutic index in the disease species. And importantly, you might be able to identify biomarkers of either safety or activity in that model that you can then take forward into the clinical trial that you can help monitor either activity or toxicity. And importantly, the increased sensitivity actually might be a good thing. I mean, it's not entirely clear that a normal volunteer predicts better to a diseased human than a diseased animal predicts to a diseased human. We don't know that. And again, the idea being that the animal model might help us get to the dose. So the case-by-case approach, which is not did you do, it's question-based, really came about in the late 80s and the 90s with the biopharmaceuticals, where traditional principles were relevant, but the practices became perhaps not so relevant. Importantly, this is not a minimalist approach. This is not the least amount I have to do, but it's a rationalization of what's the best way, what's the best model, how best can I extrapolate, how best can I, understand the clinical questions of concern, and try to mimic that as best preclinically. So the practices are different, but the principles are the same. But predictive value, I often refer to predictive value as predicting whether or not the FDA will accept your package, right? But it's not the case. I mean, it's really focused on the clinical, but it's less easy to predict whether or not it's going to be acceptable. Okay, because there isn't a checklist. You have to go forward and make your case. Which means that you need to have a dialogue. And my definition of dialogue is dialogue does not equal monologue plus monologue. I mean, it means that you need to talk to the regulators, which is not easy. But if you are a cell-based therapy and gene-based, so how many people are in cells? And how many people are in monoclonals? And small molecules? So I've tried to incorporate you all, so you'll have some small molecule slides. But the advantage you have in terms of the cell-based therapies is you have this option of not only a pre-IND, as Keith was talking about, but a pre-IND, which may get to the question of, providing the agency with a number of vectors or whatever that you said, and your growth factors, transcription factors, whatever you want to ask them and you're concerned about, and you can put it forward to them, and they can come back with an early, assessment or guidance in that regard. And you can design your animal studies, and you can provide them to them, and they'll give feedback. So that's huge. That's very huge the necessity to have dialogue, and they're very open to that dialogue. So why case by case? So when it started, I mean, it was the realization that even within a product class, there's differences. So all monoclonals are not created equal. Again, this has evolved over time, but you can have whole molecule, you can have fragments. You know, back in the day, early days, they were murine and then chimeric, and now there's humanized and fully humanized. You can have different isotypes, you can have different cell substrates, you can have mono-specific, bispecific. I've worked on a tri-specific antibody. You can have naked or conjugated. They can be uniquely specific to, let's say, a tumor, or they can be broadly non-specific, etc. And the function can be an agonist, antagonist. So this is the reason why case by case, is because based upon these product attributes, we would design our studies to try to understand safety with respect to product attributes, which is the same thing for cell therapies. I mean, they're not all created. You know, the cell source, the heterogeneity of the product, again, the differentiation or the stemness of the product, you know, the degree of foreignness. Is it autologous? Is it allogeneic? Is it xenogenic? Okay. So even within cell therapy, this is the reason why there isn't anything specific of exactly what I have to do, because you have to understand your product attributes to help inform how you design the study. So again, this is for our small molecule and monoclonal as well. I've tried to kind of explain a little bit in this table form about what I meant by product attributes and the considerations. And so if you're looking at manufacturing, obviously biological cells and tissue for cell-based biological synthesis and chemical synthesis right here. Cell-based therapy is more heterogeneous, as well as biopharmaceuticals, but pharmaceuticals are more homogeneous. And impurities are difficult to qualify or sometimes even quantify, but they're easy to qualify in small molecules. And the delivery system, you know, with cell-based therapies, we might have a device. It might be really novel route of administration. This is really the formulation, actually, and the delivery. And for pharmaceuticals, the formulation is more complex. The half-life, again, we're talking about a single dose, perhaps, which might be lifetime, versus days to months for a recombinant protein or a monoclonal antibody, and minutes to hours when we're talking about a small molecule. So again, and the toxicity is generally related to the mechanism of action when we talk about biopharmaceuticals. And certainly with our cell therapies, there's this complexity about host response for a xenogenic tissue. For our biopharmaceuticals, it's generally exaggerated pharmacology based upon the mechanism. And for the pharmaceutical, it's often unpredictable. And then you have to deal with metabolites. So that's what I mean by product attribute. When you think about that, that helps you in terms of the design of your study. Now, with respect to the principle, okay, so again, we're testing a test article, right? So we have a test article across the board. For a pharmaceutical, you're always testing the clinical candidate. For a biopharmaceutical, we might test the clinical candidate, but we might have to test a surrogate antibody, a homologous protein, right? And for cell-based therapy, we ideally try the clinical candidate, but in some instances, we do look at the analogous product to help us understand mechanism, etc. Again, PK, ADME, cell migration, biodistribution, trafficking, and that's the same as PK, ADME for cells. And then for biopharmaceuticals, we don't really do ADME. We do PK, and we might do absorption. Again, they're catabolized, not metabolized. So the principles of where does it go are the same. It's just we're looking at it. The practices, how we evaluate are different. And again, the dose levels we talk about in terms of a maximum feasible dose, or we're not sure in terms of cell therapies how best to extrapolate dose, whether or not it's a body surface area or a body weight or, a target tissue. Clearly for small molecules, we have a very good algorithm for that. And for large molecules, generally above 100,000, we extrapolate based on body weight, and less than that, body surface area. So again, We still need to extrapolate. We do it differently with different molecules. So most are in cell therapy, it looked like, and we have some unique considerations in stem cell products. Issues in terms of ectopic tissue formation, tumorigenicity, and oncogenicity, and probably tumorigenicity is the big one, right? And of course, the differences between small molecules is carcinogenicity studies to assess potential proliferation, carcinogenic, tumorigenicity, et cetera, is actually conducted in phase 3 to support product approval. But for cell therapies, we have to answer that question before we get into the clinic. And so we can talk about designs of that and challenges in terms of the tumorigenicity. So case by case. So then the other message is to ask the question. So focus on your studies by basically asking questions, not doing studies, okay? So we ask, what's the optimal procedure to get, we'll use cells in this case, cells in, what's the route, the anatomical site. So, and then what's the timing? Is it, if it's a disease model, if it's an injury model, do we worry about when that timing is? So what is that window of opportunity to treat? And where does the product go? Okay, that's your PK ADME. Will you need another dose? Will you need chronic immunosuppression? And again, our animal models will generally all be either chemically or genetically immunosuppressed, but the question you'll ask is, how does that now translate into the clinic, and will we need suppression, chronic immunosuppression in the clinic? And is there any potential to see activity in those early clinical trials? And is the population a disease population and a vulnerable population? Are you going into kids for first in human? So ideally, you conduct in one or more relevant species. The reason why you do two, it helps in the extrapolation to dose, right? If you have two, you can put them on some type of a graph and try to extrapolate to the clinic. I had a client once say, well, the flow pattern in the monkey brain is more similar to humans. Okay, we're going to do a rat study. Do you think FDA will make us do a monkey? Okay, I'm going to ask you that question again. The flow pattern in the monkey brain is more similar to humans. Okay, how many people would do a monkey study? How many people would care what the FDA said? So if the FDA said no, would you not do it? Again, what do you want to do? You want to find a clinical dose, right? So basically my answer was, even if the FDA said no, I would do it, because you want to find the clinical dose. So you want to use a test article that's representative of the clinical material, and I think we can talk about the challenges of using autologous cells, et cetera, but it's representative. I wouldn't get too crazy about talking about comparability early on, because this is the opportunity to understand and characterize the widest range for your molecule, your cell process, whatever. So I think we need to talk about in terms of, so I think the term representative is a better term. Again, ideally you use your clinical material, but if not, you will then have to assess your analogous product or homologous or whatever the molecule is. Again, for the route of administration, you might need another species just to address if you have a large clinical device. So you might be able to do most of the studies in a rodent, but you need to still address the question about any toxicity or local reactogenicity with the clinical device. Then that would kick you up to at least doing some type of assessment in larger animals. And then provide a safety margin for the clinic. So you need to know your test article. You need to know characterization of your cellular products. So for cell-based therapies, again, you need to know how best to deliver and where the cells go, the fate of the cells. And then what if the toxicity of the cells? Is it off-target, on-target? You need to understand that. Are there any impact of any drugs on safety and efficacy? And then, again, we think about the population because we are going into an index population. So the challenges for cell-based therapies are that we're looking at concentrations and volumes, location of injections, the number of injections. Keith mentioned cell stability. So these are all in terms of dose administration that we don't think about in terms of, let's say, small molecule delivery. So how do you extrapolate the dose? Again, we don't have a good algorithm for dose extrapolation. The expanded cells are encapsulated cells. The scaffold, if cells are in a sheet, how do we best extrapolate cells? And so we have multiple scaling factors. And then if you have the opportunity to have more than one species, you could do some cross-species validation. So again, there are no guidance out there. There won't be any FDA guidance to say this is how we extrapolate, that you will put forth a case based upon the totality of your information on what is the best way that you see based upon whatever studies you have, in vitro, animal studies, etc., on how I'm going to scale to that dose, how I'm going to pick that clinical dose. So again, so some of the design considerations, so this is the practice. Again, there might be cases where we might ask some questions in normal animals, and most often we do disease models and consideration of whether or not we want a placebo or a sham or a positive control and the availability of positive controls. And then, again, as I said, you want to mimic the clinical treatment. So if there is a device, you want to understand how the cells are delivered by that device. Again, this timing, if it's a surgical intervention, the window of opportunity is important to understand. And then over the course, you would want to understand interim and a term and then a recovery. So you want to have some understanding of, again, early time points and late time points. And then how big is big? Okay. More than 2. Okay. So for smaller animals, you know, you're generally up at least 5 and 10 per time point when you're, and generally both sexes, unless there's some justification to use only one sex. And for larger animals, you know, you can ballpark about 4 or 6 per group. And again, the number of groups are going to be dependent upon really what your product is. And in your animal model, sometimes it's a little bit difficult if it's your efficacy model and the dose that works in your efficacy model is the maximum feasible dose. That's difficult because then you don't have the opportunity for a safety margin. Okay, so, and then that's another discussion. And again, these are numbers, but if you're into a surgical intervention, etc., you will have to actually use more animals to make sure that if there's any attrition due to the procedure, right, you need to know that these are not 4 to 6 that you start with, it's 4 to 6 that you end up with. And then you measure, again, standard toxicological endpoints, but you may include, you know, more sensitive indicators because we're concerned about proliferation and tumorigenicity with respect to proliferation. And then tumorigenicity, again, we can have a discussion in terms of how many and how long. It's going to be based upon the stemness of your cell. So in terms of tumorigenicity studies, there are some general considerations across cell types, and then there are some cell-specific considerations that you need to take into, again, based upon the proliferative capacity, cytogenetic stability, and route of administration, mode of action. So there's a number of considerations that you would consider based upon whether or not you're adult stem cell or iPSC cell or derived from ES cells. So the regulatory expectations, again, you have the opportunity for a pre-IND meeting and a pre-IND meeting and probably more pre-IND meetings. I mean, the agency, again, in this group that is evaluating cell-based therapies is very interactive. You need to complete, you need study reports. So when you're submitting the INDs, you should describe the GLP-ness. So I say it's not non-GLP. You should describe what it is. So do you have a protocol? Do you have amendments? Do you have some control of your test article? So it's much easier to, and you can do these hybrid studies. I mean, you do a hybrid safety and efficacy study, but you can do a hybrid study where you might do surgical procedures in your laboratory, but you might bring prosectors from a contract research lab to help do the necropsies, and then you can outsource the pathology to CROs. So that might be a way to take advantage of expertise within your lab, not be fully GLP compliant, but you have aspects of them that look closer to GLP. Most importantly, you need individual line listing data. I mean, the agency, whether or not you do it in your lab, wherever you do it, what the agency likes to see is information on individual animals, not summary information, or so. I mean, that's what you really need. They don't. So any lab. So if you have a manuscript, that's fine, but they want to be able to trace individual animal data. So if that can be provided in addition to the manuscript, then that should be, fine to leverage your discovery research into an IND as long as you can trace animals. And then so making the case for Casebycase, again, it's product specific, okay? So based upon your product attributes, so each one of you will have a different cell type, a different indication, et cetera, a monoclonal antibody, a small molecule. I hope that I've, at least have you now understand the importance of the question-based. So it's really the questions. I mean, it's the clinical questions. Can you answer any of the clinical questions preclinically? Again, it's judicious use of animals. I don't think more necessarily isn't better. And then it really isn't a minimalist approach. It's an approach that we can utilize animals, hopefully, to give us what we need, and that's the dose. So with that, I thank you for your attention.
I think we have time for a couple of questions, and then we'll go to a panel discussion.
Yeah, I was going to wait for the microphone. Oh, sorry. It's me again. Hi. I have a question about the genetic stability of the cells, because there's been a lot of controversy recently about mesenchymal stem cells having aneuploidies, and the fact that they've already been approved, although they seem to show up with aneuploidies when people actually look. So one big question about the genetic stability is, what does the FDA consider to be dangerous? And are they ever going to require SNP genotyping, for example? That's two questions.
Yeah. Well, again, this is actually a CMC question, so maybe, but I will try to attempt. I mean, again, I don't. The agency generally doesn't, as Keith said, put out a limit until there's a problem, and then when there's a problem, then you'll go back and have a limit. So, that's really, and it's probably not helpful for people who are developing that will try to, evaluate it and then not know what to do with the results. But and they might. I mean, if there's a problem, and then they will require it. So right now, I don't think they're requiring it, but I think if it becomes a problem, that they will. So I know it's not a very satisfying answer, but it's very difficult, because it's difficult to put numbers on everything that people need a number on, right? So, yeah, I don't know. Unless you do. I mean, do you know anything about that? Yeah. Yeah.
I had a question about scaling of dosage to animals and humans. It's not really my area, but is there a logic to this? Why it's body weight or body surface, or?
I think body surface has to do with metabolism. So, I mean, back in the day, the small molecules based upon your size in terms of metabolism. This body surface area calculation is based upon different rates of metabolism, because drugs are metabolized. For the body weight, it's based upon clearance, and it's shown that proteins greater than 100,000 K scale better based upon a body weight. But interestingly, oligonucleotides, which are smaller molecules, not 100,000, actually scale based upon better. The data have shown based upon body weight. And I don't know if really it's because of their distribution. They're more tissue, they go into the tissues. I don't know. So, but yeah.
What about cells? What do you think the best way to scale?
So cells, again, I think that, I think in terms of scaling cells, you cobble together everything you have. I mean, if there's a database, a clinical database on like MSC's, administered IV, then you kind of justify if there's any clinical data available to help you in terms of transitioning that. If it's spinal cord, I think data has, again, scaling has been based upon the area, and it depends on where you put the cells. If you're putting them into the knee, then you would scale based upon the area, going forward. So it depends on the route. Mm-hmm. And then, again, there's just not a good database out there because everybody's doing something different with different cells to actually, but that's why I said if you had 2 species versus 1, I mean, oftentimes we're scaling based upon a mouse. That's a huge jump, right? So having at least 2 species, even if it's a mouse and a rat, gives you some sense of, well, how does this scale? Is it scaling based upon a body weight? And that's all I can say. Otherwise, it's just very empirical. And the problem is we don't have time for that in the clinical programs because we don't have the luxury of dosing many cohorts. So that's why, having more than 1 animal is at least better than, one is, and in just the regard for the scaling piece of it, I think is helpful.